Thank you for waiting and welcome to this online event from Pearson. Before we get started, I'd like to go over some few housekeeping items so you will know how to participate in today's event. Before we discuss how to download any associated materials and use the platform, I would like to inform you that this session will be recorded for regulatory purposes and for our quality assurance. You can download the delegate materials for this session by clicking the resource button located at the lower right part of the Zoom menu option. You can interact with the presenter and other attendees anytime by typing your questions into the chat box, which is located at the bottom right side of your screen. Please note that anything you type into this window can immediately be seen by other delegates in the room. To enter a question or a comment, type into the bottom section of the chat window and hit enter on your keyboard. You may also use the same chat box for assistance in case you encounter any technical issues during the event. At this time, everyone is joined on mute, but will have the control to unmute themselves, so please stay on mute if you don't need to speak. To give your questions, click on raise hand at the menu bar at the bottom. If you don't see this, click on the icon labeled participants and select raise hand. Otherwise, try holding Alt and letter Y on your keyboard. That's all I need to go through for now. Our presenter for today is Jillian Slater, and it's with great pleasure that I hand you over to her now. Thank you. Please go ahead.
Good morning, everybody. I'm Jill Slater, and I'm going to be giving this presentation this morning. We will move on to look at the agenda. This is for this part of the training. It introduces the important aspects of the course, focusing on specification and planning to teach the course. There's also an overview of assessment and support available from Pearson, and this part of the training is planned to last for 2 hours. The aims and objectives are to understand the assessment objectives for the qualification, understand the question types for the qualification, understand the mark schemes, practice using the mark schemes using exemplar student work, and we will spend quite a bit of time on that, and then learn about the support provided by Pearson around assessment and exemplars. Now, welcome to Pearson. Pearson Edexcel are the world's leading learning company and the UK's largest awarding organisation, best placed to provide qualifications aligned to the British educational system. Our international heritage stretches back over 150 years. And today we partner with schools, universities and employers worldwide, offering world-class, globally recognised qualifications to over 3.5 million students a year. Now we're going to have a quick overview of the specification. It's a modular specification divided into IAS and IAL. The total qualification is a total of 6 externally assessed units or modules. Units 1, 2 and 3, which you can see now, comprise the IAS. The content of these 3 units provides students with a grounding for A-level studies. Units 1 and 2 are each split into 2 main topics and themes. So you can see Unit 1 is molecules, diet, transport and health. So that is subdivided into molecules, transport and health is topic 1. And then topic 2 is membranes, proteins, DNA and gene expression. If we move on to look at the second part of the course, the IA2, this would be taught in the second year of study, units 4, 5 and 6. And the content of these units is focused around developing a good understanding of fundamental biological processes. These are the more demanding biological processes. And again, units 4 and 5 are split into two main themes. In both the IAS and IA2, Unit 3 and Unit 6 are the practical skills. In Unit 3, it will be practical skills just from topics 1 and 2. But in Unit 6, it can be experimental skills acquired from all 4 units, 1, 2, 4, and 5. We're going to go on from there to look at the assessment objectives. I'll just keep an eye on the chat box at the same time. Yes, I don't. That's fine. There are 3 assessment objectives, and they describe the type of learning that is being assessed. So A01, assessment objective 1, is to demonstrate knowledge and understanding of science. Questions matching this assessment objective will require students to demonstrate learning of biological knowledge and understanding, which is detailed in the specification. So anything in A01 is from the specification. A02A is the application of knowledge and understanding of science in familiar and unfamiliar contexts. So questions matching this assessment objective will require students to apply their biological knowledge and understanding. It will often involve the application of learning to a novel or unfamiliar context. A02B is analysis and evaluation of scientific information to make judgments and reach conclusions. So questions matching this assessment objective will require students to use their biological knowledge and understanding to make sense of information provided in the examination. It will often involve the application of learning to a novel or unfamiliar context. A03 is experimental skills in science, including analysis and evaluation of data and methods. Any question that assesses practical skills will match this assessment objective. So we're going to look at where each of the assessment objectives are assessed. The assessments for the qualification must match the specified assessment objectives target. So when the paper's drawn up, apart from checking for the content, it's checked to make sure that the assessment objectives are correctly distributed. The main assessment objectives for Unit 1, 2, 4, and 5 are A01 and A02A. These assessment units also have an element of A02B. A03 will be exclusively assessed in the units 3 and 6 examinations. For the unit 1 and 2 exams, approximately 35 out of 80 marks will be A01 and the same for A02. The remaining 10 marks, A02A, the remaining 10 marks will be A02B. For the Unit 4 and 5 examinations, the A01, A02A balance shifts a little, with approximately 34 out of 90 marks for A01 and 39 out of 90 marks for A02. The remaining 17 marks are A02B. We're now going to go on to think about the command words. Students should pay particular attention to the command words used in questions. These command words will identify the type of response required. For example, two common command words are describe and explain. Describe the cardiac cycle and explain the need for a cardiac cycle would require quite different responses. If you look at Appendix 7, Taxonomy, on page 68 in the specification, that lists the vocabulary or command words that will be used in questions and what is required to answer questions with those command words. Sometimes the command word identifies the assessment objective. However, usually the command words need to be interpreted in the context of the question or alongside the specification to identify the relevant assessment objective. This slide shows how different command words have been used to assess different assessment objectives. Some of the command words, those in red, will almost always be assessing A01, e.g. compare and contrast, complete, draw, state, name. Some will be generally associated with AO2A. Those are the ones in green, such as calculate, determine, show. Others will be largely restricted to AO2B, those in blue, assess, comment, criticize, discuss, and evaluate. However, a small group of frequently used command words will be used across all the assessment objectives. These are describe, explain, and suggest. In the practical skills papers, Unit 3 and 6, any command word can refer to AO3. If you look at AO1, it says that most multiple choice questions will be AO1. There are 10% of multiple choice questions on each paper. They will mainly be AO1, but not entirely. Some of them will test other assessment objectives. So now we're going to have a look at how you can map assessment objectives, and the questions that we're going to look at throughout this presentation are all in the sample assessment materials on paper WBI11, which you can access from Edexcel website. If you're going back through this presentation afterwards, you would probably benefit from matching it to that question paper. So if we have a look at these questions, state what is meant by osmosis, explain why oxygen molecules can pass directly through the cell membrane, calculate the volume of blood, explain the difference between the dissociation curves, explain why the codon for the DNA genetic code must contain at least 3 bases, explain how a particular mutation, which is given on the paper, results in a non-functioning CFTR protein. So I won't ask you to enter next time I'll ask you to enter them in the chat box, but can you just spend a couple of minutes thinking about which assessment objective you think each one matches, and then we'll have a look at what they were matched to when they were written. And I've just seen there's a question in the chat box about differentiating between what's required from a comment and an evaluate question. To some extent, it depends on the context of the question. A lot of these questions, particularly comment and evaluate, require an analysis of data, either experimental data or an experimental technique, or an unfamiliar context of biological material. And the question needs to be read in the context of what is given on the exam paper. I hope that helps. If we look at these assessment objectives, right, state what is meant by osmosis is assessment objective 1, because osmosis is a specification term. Candidates should be able to recall the meaning of the term. State is almost always used for AO1. Explain why oxygen molecules can pass directly through the cell membrane. That is 2A, because candidates should know the structure of the cell membrane, but they're being asked to apply this knowledge to explain why oxygen is able to pass through the membrane. Calculate the volume of blood is 2A. It's a calculation using data provided and is considered to be an application of knowledge, and therefore AO2A. Calculate questions will, in general, be AO2A. Explain the difference between the dissociation curves, and these are dissociation curves that are given on the question paper. Candidates are being asked to explain them. This is considered analysis and evaluation of scientific information, so it is AO2B. Explain why each codon for the DNA genetic code must contain at least 3 bases. Although this is an explain question, it is AO1, because from the specification, candidates are required to understand the nature of the genetic code. So in answering this question, candidates are demonstrating their understanding, so the correct assessment objective is AO1. Explain how a mutation that is described in the question results in a non-functioning CFTR protein. That is assessment objective 2A, because candidates need to apply different specification points to provide an answer to this question. So they're taking their knowledge from different places and putting it together. We now have some more questions, which I would like you to have a look at, and I will give you a few minutes if you could put what you think are the assessment objectives into the chat box. We will have a look at all of them and then we'll go through them all together. You put what you think they are into the chat box and then we'll talk about them. We've got quite a few in the chat box. I'll give it just another minute to let a few more people put their ideas in. Right, we'll have a look at it. Most of you put in, I think all of you put it in that state what is meant by the term phenotype is assessment objective 1. Phenotype is a specification term and candidates should be able to recall the meaning of the term. And as we've said, state is almost always AO1. The next question, calculate how many times bigger is 2A. Calculations are considered to be an application of knowledge and therefore 2A. Explain how the appearance of, I won't try and pronounce that, show it is adapted to its habitat. And again, this is using information that will be in the question. So it's 2A. Candidates are applying their biological understanding of adaptation using information provided to them. They're given a prokaryotic cell and asked to draw 3 labeled features on the diagram. AO1 candidates are expected to know the structure of bacteria, and by drawing these structures, they're demonstrating their understanding. So the correct assessment objective is AO1. The next question, explain the distribution of bacteria in the digestive system. Use the information in the diagram and table to support your answer. This covers 2A and 2B. They are both applying their biological understanding and using this to interpret information provided in the question. And then the final, describe the role of the rough endoplasmic reticulum, is a straightforward application of biological knowledge, so it is AO1. We're now going to come on to think about the languaging questions. It is really important that candidates read the question carefully. Many candidates produce answers that have got lots of correct biology, but gain relatively few marks because they're not answering the question being asked. And a significant reason for this is that candidates do not read the question carefully. As well as the command words, candidates need to consider the context of the question. Are they being asked to explain how something happens or why it happens? Are they being asked to calculate a percentage increase or a percentage decrease? Is the question asking about a particular example, e.g. this protein, these enzymes? Now, if you look at these two answers, the question is anticoagulants, antiplatelets, and thrombolytics are drugs used to treat blood clots. One anticoagulant binds to the active site of thrombin. Explain how this drug reduces blood clotting. So they're being asked to explain something. There's 2 marks of this question. Response A wouldn't gain any marks because although what they've put might be correct in some circumstances, they've not read the question carefully and have explained how a different anticoagulant would reduce clotting. Response B gains 2 marks because it's used the information given to say that the anticoagulant stops thrombin binding to fibrinogen, so fibrinogen cannot be converted to fibrin. That just shows how important it is to read the question carefully. Now we're going to think about the mark scheme and how the mark scheme is drawn up. Mark schemes describe how a paper is to be marked. The mark scheme identifies the marks for each question. It's produced at the same time as the paper. However, it's adjusted after candidates have sat the exam, account for the sort of responses that they are producing. It's not changed significantly, it is adjusted. The mark scheme for the majority of questions in IAL biology papers are point-based. This means there are independent marking points for which a mark can be awarded. In general, for a points-based mark scheme, the number of marking points will be equal to, or possibly one more than the number of marks available. So for a question worth 4 marks, there will be 4 or possibly 5 mark points. In each of the unit 1, 2, 4, and 5 examination papers, there will also be 1 or 2 levels-based questions. The mark schemes for levels-based questions are different, and we will look at an example later. So we're going to look at using the mark scheme. The mark scheme provides the answer for a question. Questions are marked by experts who interpret and apply the mark scheme to candidates' responses. There may be a number of options for a particular marking point, and some guidance is provided as to what alternative wording is acceptable. Each marking point is a standalone mark. Candidates don't need to gain the first mark in order to gain the second mark, and so on. So this is an example of a points-based mark scheme to a question. It's a fairly typical mark scheme. The question is worth 3 marks, and there are 3 marking points. So the first column gives the question number, identifies the question. The answer is provided in the second column. Each marking point is given as a separate bullet point. Additional guidance for each marking point is provided in the third column. Note that 2 types of additional guidance are provided: ignore and accept. These are self-explanatory, but in this example, chemicals would not be accepted in place of enzymes. In contrast, in the second marking point, thickens or becomes impermeable would be accepted as alternatives to hardens for the zona pellucida. But zona pellucida is the only term that is acceptable for that part of the mark. Now, if we have a look more closely at the first marking point in this mark scheme, these conventions with brackets apply to any A-level mark scheme. Now, the first thing to be aware of is that the marking point is fairly substantial. To gain the mark, the candidate needs to address the full marking point. So they must mention cortical granules fusing with membranes and release of enzymes to gain the mark. And this is characteristic of mark schemes in the new IAL qualification. Now, if we have a look at the use of brackets in the answer box, curly brackets identify alternatives, one of which must be present. So in this case, candidates must refer to cortical granules or cortical vesicles. Smooth brackets identify contextual information that may be omitted in the candidate's response but should not be contradicted. So the mark would be awarded if candidates described cortical granules fusing with the cell membrane and releasing enzymes. However, if they suggested that the granules fuse with the sperm cell membrane, the context is wrong and the mark would not be awarded. And that just exemplifies that. Now, if we look at the level-based mark schemes, a number of 6-mark questions, there are always 6-mark questions, not fewer than that. And there will be at least 1 and sometimes 2 on each of the papers for units 1, 2, 4, and 5. And these the mark schemes for a level-based question have 2 components. First of all, they have the indicative content, which is an indication of the sort of creditworthy comments candidates might make. The indicative content that's included in an answer can be a good indication of the quality of the answer. However, depending on whether or not they've answered the question correctly, the number of indicative content comments a candidate makes and the score awarded to the response may not correlate. So we've got a question that says explain it. Again, it's based on information already given on the question paper. And it says explain what the solution of sugars should contain to preserve pineapples. Use the information in the table to support your answer. So the sort of things that might be included is that the sugar solution used has a lower water potential than the cytoplasm of the cell. The sugar solution should be hypertonic, so that water will pass out of the cytoplasm by osmosis. Concentration of the sugar solution should be the same as the cytoplasm, so that sugars will not diffuse out. And the D's and the E's refer to description and explanation. The indicative content is then used to create the second element of the level-based mark scheme. So this part of the mark scheme is the levels description. So it's divided into level 1, level 2, and level 3. And then each level is subdivided in whether 1 mark or 2 marks can be awarded. And the level descriptors describe how indicative content should be interpreted to award a particular level. The sophistication of the response increases as you go from level 1 to level 3. And the candidates have to achieve all of level 1 in order to move on to level 2, and the same all of level 2 to move on to level 3. So it's not a question of just counting up the number of indicative marks they've got. They've got to fulfill 1 level before they can move on to the next level. So we're now going to have a look at an actual mark scheme, and we are going to have a look at how some questions can be marked. And I'm going to ask you in a few minutes to be looking at some questions and marking them and putting your answers into the chat box. The questions are all in the resources pack that you've received. The mark scheme and comments will be shown in the PowerPoint, but the candidate responses are not in the PowerPoint. So we're going to consider how a mark scheme is applied, and we're going to take a look at a paper and a mark scheme. On the front of the mark scheme is the general marking guidance, and it is there for examiners. It is consistent across all mark schemes. It's a series of bullet points that examiners need to make sure they are aware of. And the guidance is designed to encourage positive marking, which rewards candidates for what they know, rather than penalising them for what they don't know. It's important that all candidates receive the same treatment. The first candidate must be marked in exactly the same way as the last, and examiners are taken through the mark scheme before they start to mark, and it is explained to them what is needed to apply the mark scheme. They should be applied positively. Examiners should mark according to the mark scheme. All marks on the mark scheme should be used appropriately. They're all designed to be awarded. Examiners should award full marks if deserved. And where some judgment is required, mark schemes will provide the principles by which marks will be awarded. Examiners all report back to team leaders, so if they are in any doubt, they've got someone to consult. The principle that Edexcel use is that any crossed out work, if it's just crossed out and nothing is written as an alternative, then it is marked. If it's crossed out and alternative response has been given, then it is the alternative response that is marked and the crossed out work is ignored. Right, so we're going to have a look at a question. Describe the conclusions that can be made about the risk factors for CVD. Use the information in the graph to support your answer. Again, this question is from the SAMS paper, and some information was given about the risk factors for CVD. There are 2 marks available, and there are 2 marking points. So have a look at the exemplar responses to this question that you've got in your pack. There are 4 responses. So if you have a look at all 4 of them, decide what marks you would award using this mark scheme, and put your answers or what mark you would give to all of them into the chat box. I'll give you a few minutes to do that. I'm looking at the question about applying the mark scheme. If the second part of an answer contradicts the first marking point, do they still get the first marking point? If they write something that totally contradicts it within the same answer, then no, they can't have the marking point. If they write smoking increases the risk of CVD, and then they put age increases the risk, but smoking decreases the risk, then they've contradicted themselves. They couldn't have the smoking mark, but they could still have the age mark. This question is less likely to happen, but there are instances where it might. What we look for is the specific statements on the mark scheme. So if they've talked about something that's not on the mark scheme, not fully related to the question, but is actually incorrect, that can be ignored as long as it doesn't contradict what the mark scheme is looking for. So, for instance, in this question, if they made the two statements, but then incorrectly went on to say the effects of smoking on the wrong part of the body or on different effect on the lungs, something that was not relevant to the question, that would be ignored because they are being asked to use the information from the graph. I hope that helps a bit. Right, can you put your answers a bit out? We've got some in the chat box now, I think. Students can write their answers in bullet points, and in actual fact, they won't be penalised necessarily for writing in bullet points in the asterisk questions, but they have to be, because they will be being asked to discuss or evaluate, it shouldn't be just a series of facts. If they just put a series of facts, they probably won't have answered the question. I'll give you a couple more minutes. Those are to put the marks in on the whole agreeing with the marks that were awarded. These are student responses, although it's a sample paper, they are responses from a paper that was set and has been answered by students and has been marked by examiners. I'm leaving Ellie, who I can see is working on it, to help those of you that are struggling to access the resources. So response A was given 2 marks for saying as the age grows, the relative risk will increase, and in every age group, the risk is higher in smokers and non-smokers. Response B also got 2 marks. Response C gets 1 mark. because it only says that smokers have a higher risk of developing CVD than non-smokers. Makes some comment about the effects of age, but it doesn't actually explain them. And again, 1 mark for the final one. This is a comparison question. You have to use the words increase or decrease or higher, not just state the risk for each group. And the next question we're going to look at is a fairly straightforward genetics question. It says rabbits can have brown or white fur. The heterozygous rabbit will have brown fur. So from that, you can conclude that brown fur is dominant. So it asks for a genetic diagram to show genotypes and phenotypes. So you need both of the baby rabbits produced if 2 heterozygote rabbits were bred together. So you have to draw a diagram. It says a diagram. Candidates have to draw a diagram. And in the diagram, they've got to show the genotypes of the parents or the alleles in the gametes, genotypes of the offspring, and then the corresponding phenotypes to match the genotypes that are given. So you have got 4 of those questions. If you could mark them again and put the marks into the chat box, please. Right, we'll have a look at the marks that were awarded for this question. Now, this question also gives an example of the positive marking that is applied. So the first response was given 3 marks. They've got parental gametes and offspring genotypes are correct in the diagram. And it was taken that it could be inferred which genotypes applied to which phenotypes from the diagram that had been drawn. So that was given all 3 marking points. The second one very clearly gets 3 marks. The third response wasn't given anything because they got the first, because they hadn't got the correct gametes, it wasn't possible to get the correct answer. So that didn't get any marks. Now the third one was given 3 marks because it's got a clear key, and although the convention is for capital for dominant and lowercase for recessive, it doesn't, that's just a convention, and because they have made it absolutely clear which genotype is linked to which convention they have used is applied to which phenotype and genotype, they were awarded the 3 marks. So now we're going to look on to a question that says a calculate question. So we've got this question carries on to use it to calculate a number of heterozygous pairs of rabbits were bred together and produced 284 baby rabbits. Calculate the number of homozygous brown rabbits, heterozygous brown rabbits, and white rabbits produced. Because this is 3 different responses, 3 different calculations with 3 different answer lines, there is no carry forward within the question. However, there is carry forward if they got incorrect ratios in the previous part. So you have got a number of responses, 2 responses. So if you could quickly put into the chat box the marks that you would give them. Fairly straightforward question, this. But I'll answer a couple, I'll move on to this question in a minute, but there was a couple of questions on the chat box. The previous question, D was given 3 because, as I said, it is a convention that we use uppercase for the dominant allele, lowercase for the recessive allele, but it is only a convention. They can actually use any letters they want, as long as they make it clear, which this one does in the key, what they are using them for. And they obviously, although they didn't the other way around to what is convention, they had the heterozygous as brown. So they were not confusing which was recessive and which was dominant. There isn't any error carried forward if they get the original gametes wrong. Yeah, and we've got lots of responses. So A gets 3 marks, is very clear, well set out. All 3 answers on the answer lines are correct. The final mark, the second question gets 1 mark because they've only got 1 answer right, and each of the answers are looked at separately when there's 3 answer lines. Right, next question that we're going to look at is about haemophilia and the inheritance of haemophilia. So candidates are being required to apply their understanding of the inheritance of red-green colour blindness, which they will have learnt about as an example of an X-linked inheritance pattern. They're given information that says haemophilia is inherited in a similar way. They're asked to explain why more males than females are affected with haemophilia A. So these are the marks that we're looking for, because the gene or allele is located on the X chromosome. The defective allele is recessive, or the allele that causes haemophilia is recessive. Males with haemophilia will only have that allele, and it gives alternatives that are acceptable in the additional guidance. So and don't forget about the curly and the smooth brackets and the importance of those. Right, you've got some responses to this question. You've got 4 responses. Could you have a look at them, decide what mark you would give them, and put that into the chat box, please? Right. But we're getting much more variation in responses this time, largely because candidates struggle to express mark point 3 clearly. I will show you the agreed marks, and then I'll come back to the mark scheme to think about so mark point, the first one was given 3 marks. Candidates often use terms such as it and they, which can be accepted as long as it's in the context of the question. So it was accepted as referring to haemophilia for mark point 1 in response A. So that got the 3 marks. B didn't get anything, there was nothing seen there. In question C, or response C, quite a few of you gave this 2 marks. Now, when it was marked, it was given all 3 marks. But mark point 3 is very borderline. Higher chance of having a healthy X. Females have a higher chance of having a healthy X. It's just about equivalent to the additional guidance for mark point 3. If we go back and have a look at that, healthy males will not carry a healthy normal alleles. Females need both defective alleles to be homozygous. That was just about acceptable for mark point 3. It is possible that some examiners would not have given it, because with candidates don't always write what you want them to write. So a judgment sometimes has to be made. As much emphasis as possible on consistency is made when examiners are being trained to apply the mark scheme and discussions around it. But there'll always be some expression that candidates will express it in such a way that it comes down to the judgment of the examiner. And then D, they wrote an awful lot, but the marks were actually given in the last 3 lines. The first mark is given on the second line, and then unless the female offspring acquires both recessive alleles, the disease is not expressed as the dominant allele suppresses it, gives it mark point 2 and mark point 3. As it says it's recessive, and it says the female has to have both alleles in order to have the disease. Again, it's not the most straightforward way of expressing it, but it does get the marks. We're going to very quickly have a look at how the multiple choice questions are set out and how the mark scheme is set out. There are 10% of the marks in each paper, 1, 2, 4, and 5 will be for multiple choice. They all follow a standard format of a question stem, followed by 4 options, only one of which is correct. And then in the mark scheme, it gives the correct answer, and then where appropriate, gives an explanation of why the other answers are not correct, just to ensure there is only one correct answer. Now, the next question we're going to look at is a compare and contrast answer. In order to gain full marks, candidates need to give both similarities and differences. You can't get full marks for a compare and contrast answer if you only give similarities or only give differences. And to ensure that this is the case, the question is written and the mark scheme constructed so that in this case, there are 2 similarities and 1 difference. It might be two differences and one similarity, or it might be two similarities and two differences, because you've still got to get one from each side in order to get full marks. So they're asked to compare and contrast the structure of amylose with the structure of a malapactin. The additional guidance to examiners is not to piece together. They have to be comparing or contrasting the two molecules. Note that polymers is in smooth brackets. It's alpha glucose that is required. Bonds or links are alternatives for the second marking point. If the alpha is applied to the glycosidic bonds, then the first marking point can be given, even if they only refer to glucose. It's quite a bit of additional guidance there. Right, I will leave the mark scheme up. Can you have a look at the responses that you've got and put into the chat box the marks that you would give them? Right, we'll have a look at the marks that were given for these. You have given quite a bit of variation here. So the first response gets 2 marks because it clearly expressed the similarities and differences in the bonding. So it got mark point 2 and mark point 3. But there's no reference to alpha glucose, so not mark point 1. And what the candidate has done is compared and contrasted the functions of the molecules, which is not what they're required to do and not answering the question. Response B, again, got 2 marks, mark point 2 and mark point 3, because it missed the alpha in front of glucose. So it didn't get mark point 1. Now, I see that it's got alpha scattered all over the place, but it's alpha, it's not linked the alpha to the glycosidic. So that was not an acceptable alternative. Response C gained all 3 marks. In this case, the benefit of the doubt was given because alpha amylose and amylopectin are polymers of alpha glucose, not alpha glucose monomers. But in the context of the question, it seemed fair to interpret the sentence as amylose and amylopectin are made from alpha glucose monomers and allow the candidate to have mark point 1. Again, it's easy to see how you might, on some occasions, might come to a different conclusion. And then response D gets 2 marks. It's got the glycosidic, the comparison of the bonds, and by saying that they've both got 1,4 glycosidic bonds, they get the similarity and the difference. Right, we're going to go on to look at a question that this is based on information that was given about an amoeba and explain how the uptake of substances would be affected if the amoeba increased its number of pseudopodia. So the answer has to have an element of explanation. So it says, what would happen if the pseudopodia were increased and why that would affect the uptake of substances? Although an increased surface area will speed up uptake, it doesn't mean that there'll be any increase in the amount of substance taken up. So for marking point 2, candidates need to be describing an increased rate of uptake or faster uptake, not an increase in uptake. This is an important biological principle, and it is something that will come up from time to time. So have a look at the exemplar responses to this question and put the marks into the chat box. You put them into the chat box as you've had a look at them. I think for D, going back to the previous amylose and amylopectin, it says that amylopectin has got 1,4 and 1,6 bonds. Amylose does that have 1,4 glycosidic bond, meaning it won't be able to break down quickly. I think it was taken that they meant to write does not have 1,4 glycosidic bond. That would make the sentence make sense. And although it's not asked up for the effect of the bonding, the explanation it gives that it won't be able to break down quickly is linked to not having glycosidic bonds. So that mark was given. Right, if we have a look at the responses to the marks that were given, and I noticed that one right, now I've got in my notes, which I would agree with, that I was sent, that A, B, and C each gained 1 mark because uptake of substances or uptake would be more were not allowed for mark point 2. And in response B, it was accepted, but they were referring to an increase rather than increase in rate. A question like this, you are looking for faster increase in rate, faster rate, rather than just an increase in uptake. So I think B would have been given 1 mark, but I can see that it is a conflicting one. And D gained the 2 marks because it very clearly stated that the uptake of substances, the rate of uptake would be steeper. Surface area to volume ratio was accepted, even though the mark scheme said surface area. I do think B should only be 1 mark, not because it says it, but because it doesn't say rate. If it said it was faster, that would be fine. Now, the next question we're going to look at is a level-based question. It's identified as level-based by the asterisk. It's got 6 marks. It's using information in the table to support the answer. It would probably be worth your while after this session actually looking at, you can access the SAMS paper on Pearson's website, and if you go back through them, it would just help clarify where the marks were given. But there's some 3 types of mutations are shown, and it says explain the possible effects of these 3 types of mutation on the amino acid sequence coded for by this length of DNA. So it's a length of DNA that's shown. It's an explain question, and it's a level-based question. So the type of indicative content, you've got your 3 types of mutations: substitution, deletion, and insertion. The effects of each of those. So, for instance, substitution only affects one triplet codon, may not change the amino acid, with an example taken from the table. And again, it may result in a stop codon with an example. The effects of a deletion with an example at the end. An insertion, again, an explanation of what effect it will have with an example at the end. And students are required to use these examples to show that they are making use of the information in the table, applying unfamiliar knowledge, or applying their knowledge to an unfamiliar situation. So that is what they might comment on. And then this is how it is marked into levels. So a correct statement about mutations will get them 1 mark. If they comment on one aspect with 1 illustration, or 2 aspects commented on but no illustrations, they will get all of level 1, so that gives them 2 marks. To move on to level 3, they need the aspects commented on with corresponding illustrations. If they describe 2 aspects with both of them having illustrations, or 3 aspects but only 1 or 2 illustrations, then that gives them the first part of level 2, which is 3 marks. If they comment on 3 aspects with corresponding illustrations, that gives them all of level 2, which is 4 marks. And then for level 3, it goes up to 4 aspects with illustrations, and then 5 aspects with illustrations. So if you have a look, I will leave that up, and if you have a look at the responses that you've got, you have got 3 responses. If you have a look at them, then I'll give you a few minutes to do that. It's a bit longer to do these because they take a bit more thought, and then we will move on. We will go through it. Again, if you put your answers into the chat box. When it says illustrations, if we go back, hang on, I'm going the wrong way. So it means the examples. So a substitution, that's using their knowledge to say what a substitution is. And then from the table they're given, an illustration is that number 6 becomes A, so it would still code for leucine. If you're talking about change amino acid, e.g. number 1 becomes G, resulting in valine. The illustration for deletion is if you remove base 4 and the sequence becomes methionine, serine, phenylalanine, threonine. So those are the illustrations. I hope that helps. So those are the aspects that they might comment on. A substitution changing the amino acid is the aspect, and then the illustration is the ones that were given previously. A deletion can change the sequence, then an illustration. So it's using the information in the table to answer the question. I hope that makes it a bit clearer. So if you start to put your answers in the chat box. And the question was, explain the possible effects of these 3 types of mutation, which were substitution, deletion, and insertion, on the amino acid sequence coded for by this length of DNA. So you've got a length of DNA and you've got the amino acids it codes for, and you're being asked to use your knowledge of the 3 types of mutations to explain what effect it might have on the length of DNA. Nadia, to go back to the amylose and amylopectin, when this was marked, it was interpreted, it seemed fair to interpret the sentence as amylose and amylopectin are made from alpha glucose monomers and allow the candidate to have mark point 1. In the context of the question, it was interpreted that they should get that mark. And as you will realise, on different occasions, a different examiner might come to a different decision, but there will always be judgment made by examiners. We don't want candidates just to regurgitate textbook answers. We want them to apply their knowledge, but that does mean that there will always be a level of interpretation or judgment by examiners. Right, we will have a look at the marks that were awarded. These are the level-based questions are probably the most difficult to mark, as well as the most difficult to answer. But the breakdown of the levels helps to clarify what is needed. So the first response got 1 mark. It was level 1. It gives one relevant aspect is commented on, because it says the amino acid will not change. But that was restricted to level 1. B got 3 marks because several aspects were commented on. Substitution may still produce the same amino acid. Deletion and addition would result in different amino acids. It gives one example: substitution of GAA to GAC would change glutamic acid into aspartic acid. That is the only illustration that's given. So that is why although several aspects are commented on, that's why it's restricted to 3 marks. It's level 2, 3 marks. The final answer received 2 marks because it says it gives several aspects. It says what effect a substitution might have, what deletion and insertion might do, but it doesn't give any illustration from the table. So a number of aspects, but no illustrations. These tend to become clearer as a marker. I've done a lot of marking, and as you mark more of the question on that particular exam paper, it becomes easier to apply the mark scheme when you have seen more examples of what students have actually written, candidates have written. We've just got a couple more questions to look at. This says determine whether this person is healthy. So they're given some information. Determine is a new command word in this qualification. And the answer to a determine question must include an element which is quantitative. So they're required to use the information provided in the question to determine if a person is healthy. So the 2 marking points, the first is for determining the pulse pressure from the information that they're given. And the second is for recognizing that a difference greater than 3.75 kilopascals, that is greater than 25% of his systolic pressure, means a person is healthy. So the first part is a calculation, and the second part is understanding what that means. And the data for the calculation is taken from a graph. Consequential error was allowed for the second marking point. So if they calculated the pulse pressure was less than 25% of the systolic, they could get in mark point 2 for stating that the person was unhealthy. So there are 3 examples for you to have a look at. If you could put the marks that you would give into the chat box. This is much more straightforward than the level-based one, so we won't spend quite as long on this. And then we've got one more question to look at. Right, going back to the amylose and the malapectin, the mark scheme says we can accept glucose if alpha was indicated when referring to the glycosidic bonds. But I think the alpha has to actually go in front of the glycosidic rather than the 1,4. Right, the marks that were awarded, the first one got both marks. They calculated it correctly and interpreted it correctly by saying they were healthy. B didn't get any marks because the pulse pressure was incorrectly calculated by giving 5.2 instead of 5.3. There's no indication of it being 25% of the systolic, which would have been allowed. But it's still, what they have calculated still means that the person would be healthy rather than unhealthy. So the statement is incorrect. And then part C, again, no marks. Incorrect calculation of the pulse pressure and incorrect interpretation of what they've got. D gained 1 mark for the correct calculation of the pulse pressure, but then interpreted that as being not healthy, so they didn't get the second mark. Now, the final question that we're going to look at, the command word for this question is suggest. So candidates are expected to use their biological knowledge in an unfamiliar or novel context. The suggest command word will be used for no more than 10% of the available marks in a paper. In the additional guidance, they ignore where molecules may be found. Because the context is unfamiliar, candidates shouldn't be penalised for suggesting measuring a molecule in an inappropriate location. So measuring carbonyl phosphate in the blood, liver, kidney, etc. would all be accepted, as would simply suggesting carbonyl phosphate without a specific location. Now, it is a bit, I apologise, but I haven't got the information that was given in the question, so I can't give it to you. I would, as I say, I would go back and just look at this with the SAMs that are in the on the website, but have a look at the marks that were given and see what you think you would give it. This is the last question that we're looking at. So if you put your marks into the chat box. Now, what the question is actually asking is to use the information in the table to explain what will be tested for, or how an inherited genetic disorder can be tested for. So you've got genetic screening, you've got a biochemical test, and you've got family history, which are the three different ways that an inherited genetic disorder can be tested for. And you can actually get the marks without naming specific tests or molecules, because candidates wouldn't be expected to have learned that information. So I can see there's quite a lot of variation in the marks that are being given. So we'll have a look at the agreed marks. Now, response A gained 1 mark because that's mark point 2 for saying the concentration of ammonia would increase. It's a named molecule that would increase. B gets all 3 marking points. Gets mark point 2 for no citrulline being produced, which is a named molecule whose levels will differ. Mark point 1 for reference to a genetic screening method, which is amniocentesis, and mark point 3 for referencing a family history. C gains mark point 3 only for genetic pedigree or family history, and D gain mark point 2 only for a high ammonia concentration. Right, I hope this has been useful to you, looking at actual examples of student marks, candidate responses, and the marks that were given. We're now coming to the final part of the presentation, where we're looking at the support that Pearson provides for you as teachers. And one of the first things is the examiner's report, which is produced for each paper. After the exam, the principal examiner considers the performance of the paper. The report will draw on attention to areas where candidates did well and those where they did not. It will include consideration of the marking points that candidates gained or did not gain for each question. And it will also be examples of candidates' responses to illustrate why particular marks were or were not awarded. So, for instance, this is an examiner's report on the performance of a cohort answering this particular question. Candidates were asked to explain the importance of the 3 inorganic ions from the graph. Nearly all candidates could explain the importance of nitrate ions. Fewer could explain the importance of calcium and magnesium. Didn't expand the explanations. And a small minority of candidates described what would occur to a plant deficient of these inorganic ions, which was not credit-worthy. These reports can be shared with students, particularly if you use peer marking or of exam papers. For each exemplar used, there'll be an examiner comment explaining how this particular response scored in the exam and will identify why marks can or can't be awarded. This is a cohort response, a cohort comment. Examiner feedback on cohort performance, this time for a level-based question. What candidates were able to do. The fact that a lot of candidates did not apply their knowledge to the given context, which will always limit the marks. You've got to relate descriptions to the given context to access levels 2 and 3, which is what we saw in the response that we looked at. Again, an examiner's comment on a particular response. And there's also an examiner's tip at the bottom. And the examiner's tip are suggestions to students that will help them prepare for a particular type of question or avoid common mistakes in answering a particular type of question. So these examiner's reports come out after the exam, and they are designed to assist with future teaching, and both when preparing your lessons and preparing your students for exams, and also allowing the students themselves to see the feedback from the exam. Now, there is support available at every stage. Free resources and support for planning, teaching and learning, exam preparation, and then results support when the exams come out. And all of these are free and available online. The web page for Pearson has 4 tabs. It's got specification, it's got sample assessment materials, which I was referring to, exam materials, which are documents including past papers, mark scheme and examiner's report. And of course, this is continually added to as more exams are sat. And teaching and learning materials. There's exemplars, lesson plans, course planner, and much more. Results Plus is support for exam preparation and post results, and it's a free online results analysis tool for teachers. It provides a detailed breakdown of student performance, identifies topics and questions where students could benefit from further learning. It benchmarks your school's performance against other Edexcel schools. But it's not just a post-results tool. Mock exams can also be fed into the system to produce analysis. You can find students' results analysis from their previous school, and it gives your students direct access to their final grades and performance breakdown wherever they are. Schools need to sign up for it, but it is free. You just sign up for a free Results Plus account. And it's linked to e-pen marking. All the exam papers are scanned. They're taken in a traditional manner. They're scanned into a system which enables examiners to mark a question at a time, and different examiners may mark different questions on the same paper. They're all marked digitally, and then it's consolidated into a central system, which is used to provide the analysis in Results Plus. Another resource you might find very useful is Exam Wizard, which is a free tool which allows you to make homework assignments, topic tests, mock exams. It uses past paper or past questions. They're tagged against the unit, topic, and assessment objective, so you can use it to create your own paper on a particular topic, drawing from all the from the bank of questions. The mark schemes are there, the examiner report is there, and it means that the most recent exam content is available sooner. Access to Scripts is another self-service online free and instant resource available from results day onwards. It allows you to see all your students' marked papers. It's available for all qualifications and is accessed through Edexcel online. The scripts don't have examiner annotation on them, but they do have the marks awarded for each question. Using Results Plus and access to scripts will help you identify topics and skills where your students could benefit from further learning. And it will also help you make decisions about EARs. Pearson Publishing supports a modular approach in line with the latest specifications, which are recognised by universities worldwide and fully comparable to UK reform and GCE A levels. It's got exam practice, it's got international content, embedded transferable skills, and it's reviewed by a language specialist to ensure the book is clear and accessible. And the final resource to help you with teaching Edexcel biology is your dedicated subject advisor, Tim, and you can contact him using the information on the slide. That brings me to the end of the presentation. Has anybody got any questions? Please tap them in chat, put them into the chat box. We've got just a couple of minutes left. Perhaps Ellie can answer the question about access to the PowerPoint. Unit 3 is experimental questions or practical skills questions using the practices, the core practicals on Unit 1 and Unit 2. And Unit 6 is also practical skills, but that one will use not only core practicals from Unit 4 and 5, but also Units 1 and 2. And standard practical techniques, use of particular apparatus, how to make results reliable, that sort of thing. Right, I will leave the meeting now. Thank you all for your attendance today, and I hope that was useful. I will leave you in the hands of Ellie to try and sort out problems with the PowerPoint. Thank you for attending. Bye-bye.
Thank you so much, Jillian, and thank you everyone for attending today's session. We hope you found it useful. Have a wonderful day and stay safe.
Thank you. Bye-bye.